The MetaHIT project was a precursor of the microbiome work of today. It led and inspired the field, basically. And that was possible because the time was right to do it. So when I conceived the MetaHIT project, I went and talked to the best scientists in the field who were interested in contributing their expertise to the project. Because everybody brought what was easy for them to do. And gathering the team, it gave a synergy which multiplied everybody's contribution to something which was unprecedented. Which I was very, very central to the project. And importantly, created tools which allowed assembly of short sequences into longer gene-sized contigs. Soon thereafter, BGI said, okay, we have assembled 3.3 million genes. So it was a little more than what we had. Great challenge is to really learn how does this organ contribute to the whole organism, right? How does this organ evolves? Can it change in different directions? Can it contribute to our health? Can it be bad for our health? I think that perhaps the most practical approach is to stay with this simple but powerful notion of the impact of loss of diversity. This we know that creates situation of risk of developing chronic diseases. And we have proposed that low richness microbiome is kind of toxic microbiome.
So I think we cannot underestimate the importance of MetaHIT, because before MetaHIT, people were thinking, you have to isolate bacteria, you have to sequence them. We're doing that again. Yeah. But still, to have the broad view, I think MetaHIT was really sort of the one way that made that possible. And I think here we also actually have to praise BGI for daring. But still, a lot of money is used on genomics rather than on metagenomics. So I still think we have some way to go to really explain why gut bacteria and other bacteria on us maybe are even more important. Of course, we know also from research at BGI, you have interaction between the genome and the metagenome, but at maximum 20%.
I mean, it was a very important eye-opener for the research community that we can study this organ in a high-throughput way with using the latest technologies. Before that, no one really believed that this was possible. That initiative actually opened up the research field that we see today is very, very important for human health.
I myself has worked for 60-some years in human genetics and genomics, and lately also in somatic cell genomics and evolution. And although I'm impressed by the heterogeneity of the human cells in our body, I realize that the metagenome, the microbiome of our different organs are even more impressive, with many more cells and especially many more interesting genes.